The cellular pool of cholesterol is monitored by the combined action of INSIG-1 or its close homologue INSIG-2 and SCAP. SCAP has a sterol sensing domain or SSD made up of 5 transmembrane alpha helices. This SSD is similar to the sterol sensing domain in HMG-CoA reductase. SCAP has 8 transmembrane alpha helices and a large C-terminal cytosolic domain. Through its C-terminal cytosolic domain, SCAP remains bound to SREBP. Both INSIG-1 or its close homologue INSIG-2 and SCAP are transmembrane proteins. Both INSIG-1 or its close homologue INSIG-2 and SCAP are located in the ER membrane. At high cholesterol level, INSIG-1 or its close homologue INSIG-2 binds to the SSD in SCAP. When the SSD in SCAP is bound to cholesterol, the SCAP binds to INSIG-1 or its close homologue INSIG-2. When INSIG-1 or its close homologue INSIG-2 is tightly bound to the SCAP cholesterol complex, it blocks the binding of SCAP to coat proteins on COP2 vesicles. Due to this blockage, the incorporation of the SCAP-SREBP complex into ER2 Golgi transport vesicles is prevented. So, SCAP-SREBP complex remains in anchored condition in the ER membrane. At low cholesterol level, INSIG-1 or INSIG-2 dissociates from SCAP. Due to this dissociation, SCAP-SREBP complex becomes free from INSIG-1 or INSIG-2. In the next step, vesicle budding occurs. In the next step, vesicular transport occurs. By this vesicular transport, freed SCAP-SREBP complex moves to Golgi cisterna. In the Golgi cisterna, enzymes like S1P and S2P sequentially cleave SREBP. These cleavages release the N-terminal BHLH domain of SREBP. This released N-terminal BHLH domain of SREBP is called as nuclear SREBP or NSREBP. The NSREBP then moves towards nucleus. In the next step, NSREBP translocates into the nucleus. In the nucleus, the promoter regions of some genes contain SREs or sterol regulatory elements. The SRE has a length of 10 base pairs. In the nucleus, the NSREBP then binds to the SREs in the promoters of some genes. By this binding, NSREBP controls the transcription of genes containing SREs in their promoters. The NSREBP activates the transcription of genes encoding LDL and HMG-CoA reductase. Regulated intramembrane proteolysis, which occurs in the Notch pathway, plays an important role in the cellular response to low cholesterol level. SREBP is a cholesterol-dependent transcription factor. Now, question arises, why does this pathway occur? If there is low level of lipids in the cell, The cell cannot make adequate amounts of membranes. If in a cell there is high level of cholesterol, then large crystals are formed. These large crystals damage cellular structures. To prevent such disastrous event, cells normally maintain appropriate lipid levels by regulating their supply and utilization of lipids. Now we will discuss about the structure of SREBP. SREBP has three different domains. They are central membrane anchoring domain, an N-terminal cytosolic domain, and a C-terminal cytosolic regulatory domain. The N-terminal cytosolic domain contains a BHLH DNA binding motif. The central membrane anchoring domain contains two transmembrane alpha helices. Now, some important points related to SREBP. Mammals express three known isoforms of SREBP. The three known isoforms of SREBP are SREBP1A, SREBP1C, and SREBP2. SREBP1A and SREBP1C are generated from alternatively spliced RNAs produced from the same gene. SREBP2 is produced from a different gene. SREBP1A, SREBP1C, SREBP2 together control the availability of cholesterol, fatty acids, the triglycerides, and phospholipids made from fatty acids. In mammalian cell, SREBP1A, SREBP1C exert a greater influence on fatty acid metabolism than on cholesterol metabolism. In mammalian cells, SREBP2 exerts a greater influence on cholesterol metabolism than on fatty acid metabolism. Please like, subscribe and share.